So, just for those of you who may not be quite familiar with heart failure, just a couple of figures to highlight the magnitude of the problem in terms of number of patients, in terms of prognosis, which remains poor, and also obviously in terms of costs. So, to make a long story short, the major underlying cause of heart failure is the loss of millions of cardiomyocytes, contractile cells, leading to a deterioration of heart function. Now, when you have a sick horse, that could be true for any other animal, you have two major options. Option one is to heal the animal, which transposed to the failing heart means to repair. Repair involves different mechanisms that you show here, and which can contribute to improve heart function. But you will note that there is nothing to do with regeneration, which would be the generation of new cardiac cells. Anyway, in this context, it makes sense to use either cells or exosomes. Exosome is a poor word. We should talk about extracellular vesicles at large, not to say secretome. Now, second option is to replace the animal, which transposed to the failing heart means triggering the generation of new cardiomyocytes. And this is the only definition for regeneration. And it is in this context that RNA therapeutics can find their place. So I think these are two major conceptual options which should be kept in mind when you're thinking in terms of therapeutic applications. Now, a brief snapshot on the landscape of cell therapy. Currently, there are two major types of cells which have been and still are investigated clinically. One hand, mesenchymal stromal cells, and there are previous talks which have addressed issues associated with these cells. And in the context of heart failure, we should acknowledge that the data so far have been conflicting. Some trials reporting positive outcomes, others negative outcomes. Second category of cells, cardiomyocytes, derived from pluripotent stem cells, either ES or iPS. Now, it's too early to draw conclusions regarding efficacy because these trials are on the way. However, in terms of safety, it's rather reassuring. So far, no red flag, and I can tell you that the first patient in whom I transplanted cardiovascular progenitors derived from ESCs is now 9 years from her surgery without any side effects that could be attributed to the cells, and this is also true for the other patients of this trial. Now, something important that we have learned from this accumulated experience with cells is that in contrast to the initial expectations that they would engraft and make new myocardium, actually, at the end of the day, and no matter what you do, so far, they die. Nevertheless, there is still some benefit, and this has led to the so-called paracrine hypothesis, whereby the primary mechanism of action of the transplanted cells is to secrete a blend of biofactors, many of which being packaged in extracellular vesicles, in which harness endogenous repair pathways, the same pathways that I have shown to you, that is, reduction of fibrosis, reduction of inflammation, stimulation of angiogenesis, and so on. So, that said, this equivalence between stem cells and their secretome has now been established in a wide variety of preclinical models spanning, as you can see, very diverse diseases. So basically, the effects of transplanting stem cells can be duplicated by the exclusive delivery of their secretome. This has been a major achievement of these past years, which raises the question, if there is functional equivalence, why would you use exosomes or EVs instead of cells? Well, the answer is that using the secretome features definite clinically relevant advantages that you see here, and probably the most important of these advantages is the lack of immunogenicity. There is a current trend to use allogenic cells, in particular those coming from pluripotent stem cells. Obviously, these cells will trigger an immune response requiring immunosuppression with all the side effects we're aware of. Well, if you take the secretome, in particular of cardiovascular progenitor cells, definitely it's not immunogenic, which represents a major advantage for the patient. This explains why, so far, you really have a bunch of clinical trials ongoing with the use of secretome coming from different cells, largely MSCs, but not exclusively. And now there is another one, which is our secret HF trial that we have started now 6 months ago. And basically, just to give you a brief outline, we start from IPS. The IPS are clinical grade, obviously IPS, are differentiated into clinical grade cardiovascular progenitor cells, and then we collect, purify, filter, concentrate the secretome, which is injected intravenously 3 times, separate by 3 weeks, in patients with non-ischemic cardiomyopathies. We've treated the first patient 6 months ago. Everything has gone well so far, and the second patient will be treated next week. So, cells and exosomes share a couple of issues, some of them being listed here. I will not detail them because I just want to highlight which, for us in the clinics, probably is the major bottleneck. And the major bottleneck is delivery. There are 2 ways of delivering cells or exosome, and I don't think that so far we've been very successful in managing delivery very well. The first option is to directly deliver the therapeutics, either the cells or the exosomes, in the organ, okay? The advantage is that you definitely hit the target. The problem is, to do that for the heart, you need an invasive approach, either a surgical open chest approach, which makes me happy, but it's not very easy for the patient, or a catheter-based endocardial approach. But in the 2 situations, you have an invasive route, which means that it's going to be tricky to repeat the procedure, whereas these patients definitely probably need to be redosed from time to time because it's a chronic disease. Now, the second option, which is by far much more attractive, is the IV approach. It's easy, it's non-invasive, it can be repeated, it's not expensive. But then the mechanism is not that easy to understand, and we come to the kiss and fly hypothesis. Kiss and fly meaning that the current hypothesis is that once you have injected IV, either the cells or the secretome, it's basically trapped in the spleen, largely, but primarily in the liver and also in the lungs, where it might shift the phenotype of the endogenous immune cells toward a reparative phenotype, and then these reprogrammed immune cells then traffic in the blood flow and hit the target organ. This is how we can explain the fact, in particular in the brain, but also in the heart, when you inject intravenously, the therapeutics never reach the heart, but nevertheless, you have some benefits. But definitely, these hypotheses need to be further characterized. And this is major practical implications. Now, the second option is to regenerate, really, that is to trigger the production of new cardiomyocytes. And once again, neither the cells nor the exosomes so far have been able to induce a true regeneration. Now, the problem here, where RNA therapeutics might find their place, the problem here is that the regenerative capacity of the heart is very variable depending on the species, and unfortunately, in the human adult heart, which is the only one we're interested in, the regenerative capacity is lost. Indeed, it's not fully true. There is a very slow turnover over life, but not enough to compensate for the loss of cardiomyocytes, which underlie the development of heart failure. But the mechanisms, and this is the important point, the mechanisms are there, as shown by this very unique observation of a neonate who suffered a myocardial infarction at birth and completely recovered without any functional defect, without any residual fibrosis. Unfortunately, the time window is very short, a couple of hours or days after birth. After that, the mechanisms in play for cardiomyocyte proliferation are just shut off. And the big issue is, now that we know the mechanisms, how can we reawake them and trigger again, in a controllable fashion, a proliferation of cardiomyocytes? And here comes the benefits of COVID. Why? Well, because COVID is based on the use of LNPs, encaging mRNA, coding in this particular case for spike, okay? The idea has come, why don't we use this technology but to put an mRNA coding for one of these factors able to reinduce cardiomyocyte proliferation. And actually, proof of principle has now been brought, in particular by this recent study, where they did exactly that, taking mRNA, coding for a factor known to induce cardiomyocyte proliferation in a large animal model, and showing definitely an improvement in function along with proliferation of new cardiomyocytes and a reduction in infarct size. So we're lucky to have been granted by the European Commission now to leverage this mRNA technology to induce cardiomyocyte proliferation by the appropriate identification of the factors able to induce such proliferation and putting them in appropriately targeted LNPs. Just to mention, however, that the issue is not trivial and that we should make clear distinctions between these technologies applied to vaccines and those applied for, say, regeneration. And again, a major difference and a major issue that we have to tackle is delivery. Because here, you have this issue of delivery at two levels. First, if you inject intravenously the LNPs, they have to target the heart and bypass the liver, which is very difficult to achieve. And number two, once they are in the heart, you have to get the expression specifically in cardiomyocytes and not in the other cell types. So you see the challenges of delivery are really there, and one of the objectives of the consortium I just showed is to try to address these bottlenecks. If we look at the landscape of RNA therapeutics right now, most of the studies actually are using microRNAs as biomarkers. There is a single clinical investigational interventional trial testing an ASO for reducing remodeling, which is enlargement of the ventricle, but not really regeneration. So to conclude, the title of the presentation was Cell Exosome RNA. I don't have the answer, obviously. But as you have seen, there are different criteria which can be taken into consideration. First is the objective, repair versus regeneration, and they're not synonymous. Number two, obviously, the soundness of the preclinical data, but also, and maybe more importantly, the manufacturing considerations. Because if we don't appropriately address these manufacturing considerations from the very beginning, and in conjunction with the regulatory authorities, it's unrealistic to figure out that the basic concept will be ever moved towards clinical applications and patient care, which at the end is obviously our only objective. Thank you very much.
Do you have some questions? Yes? Thank you very much, Professor. Do you have any question for Professor Menasché about his presentation? Yes, Professor Fischer.
Thank you, Philippe. First question. The secretome, that looks interesting, but I would be curious to know what are the molecules that are present, and even more important, among the many molecules that are present, which ones are the relevant ones to regenerate the heart cells? And could it be that based on such studies you could design a more focused study where we use a limited number of molecules?
No? 1, 2?
Okay, so easy to answer the first one. Obviously, for regulatory reasons, we had to map the full cargo, so we know exactly the type and the number of proteins on board. I can tell you we have more than 2,000 proteins on board, and we also know the microRNAs. So that's just, you know, the list of the components. Now, second question I cannot answer because the fact that a protein or a microRNA is overexpressed doesn't necessarily mean that it's the most bioactive, okay? And so when you look at the literature, it's interesting to see that each study highlights one protein or one microRNA, and it's never the same. So I'm not sure that you will be able to funnel the composition to a very limited number of ingredients which could then make a synthetic drug, because this is obviously the hope of the pharmas. So it might be the case, maybe in artificial intelligence will allow that, but so far the number of components is so huge, and the relations between all these components are so complex, the matrix is so complex that so far, nobody has really been able to say that this is the component or the group of components which account for the protective effects of the whole secretome.
So you mentioned cells, exosomes, and RNAs, not DNA. So I have a question about DNA. I know with the guy Eric Olson in Dallas, who has published recently beautiful paper where he's using base editing to modify the calmodulin kinase, I don't remember which one, in cardiomyocyte, and apparently in animal models, of course, it has a beneficial effect to control heart failure. So I wonder whether you have an opinion on this alternative strategy.
Okay, well, I'm not an expert in DNA, and I just this was the title I had RNA, so I just talk about RNA.
The criticism is just...
No. This is another option. Obviously, it adds an additional layer of complexity because then you're starting to play with the genome, okay, which is not the case, obviously, with mRNAs. But it's true that there are a couple of studies now showing that some modifications of the DNA and of the genome of the cells might trigger regeneration. My very humble opinion is that we're pretty far from clinical applications in this context. In contrast to mRNA, because with mRNA you have the bricks already there. You have the LNPs, you know how to synthesize mRNAs. And if we can address the very crucial issue of targeting, then we're not that far from clinical applications, I think.
Other questions? Yes, Marc Bonneville?
I perhaps missed something, but when you say, for instance, we are not so far when using RNA, it would be RNA coding for what?
RNA?
coding for what?
Oh, okay. So this is not the most difficult because the factors known to induce cardiomyocyte proliferation are pretty well known.
Okay. So
And so, for example, within our consortium, the European consortium, we already have the candidates.
Okay. So it would be true.
Thank you very much. Thank you. Thank you very much, Professor.